Photo-Respiration. This topic explains the concept of photo-respiration, its process, and its importance. Plants consist of leaves. If we closely observe a leaf, it is made up of many cells. Let us have a look at the single cell of a leaf. Inside the cell cytoplasm, we can see organelles like mitochondria, peroxisome, and chloroplast, which are involved in the process of photorespiration. Let us look inside the chloroplast, where the process of photosynthesis begins. Here, in the presence of high concentration of O2 and low concentration of CO2, RUBP is oxygenated with the help of an enzyme, and it forms phosphoglyceric acid 3C and phosphoglycolate 2C, which are the first products of the process. Phosphoglycolate is dephosphorylated by an enzyme, releasing a phosphate molecule and forming glycolate. Returning back to the normal cell view, we can see that the glycolate diffuses out of the chloroplast and enters the peroxisome. Inside the peroxisome, glycolate is oxidized by an enzyme to form glyoxylate. With the help of another enzyme, glyoxylate forms glycine 2C molecules. As we can see from the general view of the cell, these glycine molecules diffuse out of the peroxisome and enter the mitochondria. Inside the mitochondria, two glycine molecules unite with each other to form serine 3C and release CO2. In the cell view, we can see that serine diffuses out of the mitochondria and enters the peroxisome again. Inside the peroxisome, with the help of an enzyme, serine gets converted into glycerate. Getting back to the general cell view, we can see that glycerate diffuses out of the peroxisome and enters the chloroplast. Inside the chloroplast, glycerate is phosphorylated by an enzyme and forms phosphoglyceric acid. As a recap, we can see that due to excess amounts of oxygen in the chloroplast, the oxygenation of RUBP takes place that releases glycolate, which enters the peroxisome. Here, glycolate further oxidizes and releases glycine, which enters the mitochondria. In mitochondria, glycine forms serine and releases CO2. Serine then enters the peroxisome and forms glycerate, which in turn enters the chloroplast. Here, glycerate produces phosphoglyceric acid, which is further utilized in Calvin's cycle to prepare food. We can now define photorespiration as a respiration process where the atmospheric O2 is utilized and CO2 is released as an outcome. The process occurs in the presence of light, therefore it is termed as photorespiration.